Hey team, Jack here. So in the last video we covered how does your immune system detect SARS-CoV2 and then what does it do after it detects, which is signal. It sends a signal out to initiate the immune response to SARS-CoV2. Now we're going to look at how your body actually responds. Once it's received those signals, what does your body do, particularly your innate immune system do, in response to SARS-CoV2 after that initial signaling has occurred. So let's jump into it. Yeah, we're looking at the innate immune response after the signaling has occurred. So the first response is a vascular response. Now, blood vessels have an endothelial lining on the inside of the blood vessel, and then they're surrounded by smooth muscle, which controls the diameter of the blood vessel. This is arteries and arterioles. So blood flows down there. Look at that animation. Jeepers, James Cameron, eat your heart out. Now, on the surface of these endothelial cells, we have cytokine receptors, such as the interleukin-1 receptor 1. And that's going to receive the signal from that interleukin-1 molecule we talked about before. Now, upon receiving that signal, the endothelial cells will release nitric oxide, which acts to relax those muscles, right? So it's an inducible enzyme that produces nitric oxide, and nitric oxide is released from those endothelial cells. It will act on those smooth muscle cells, and that will cause relaxation of those muscle cells. Now, on the smooth muscle cells themselves, there is a prostaglandin receptor, and activation of that receptor will cause direct relaxation of those smooth muscle cells. And so if you activate those, you lead to vasodilation. So the space between the blood vessels will dilate and increase because the muscles are relaxing. So now more onto the capillary end of the scale. So now there is no smooth muscle around there onto the capillaries, and there's more just a thin layer of endothelial cells. The activation of these receptors causes a breakdown in the bonds between these endothelial cells, creating gaps in the blood vessels, right? So the tight junctions between the endothelial cells that make a blood vessel semi-waterproof breaks down. So now compounds and fluids can flow straight out of the blood vessels. So the tight junctions break down. Why does it become leaky? Well, it lets humoral immunity get into the tissue. So those antibodies that might be produced can get into the tissue, as well as complement proteins, which are antimicrobial antipathogen proteins that exist in your blood as part of the innate immune system can get in. The other thing that happens upon activation of these cytokine receptors is the expression of adhesion molecules. So the endothelial cells will now shoot out an adhesion molecule, and it will be expressed on the surface of that endothelial cell. And now an immune cell, like a neutrophil, can come along and those adhesion molecules will now stick. So instead of flowing straight down the blood vessel, the neutrophil will now stick. It will slowly grab onto as many adhesion molecules as it can. This will slow it right down until eventually it sticks onto the blood vessel wall. And from here, it can now squeeze through those gaps in the blood vessels and migrate towards the tissue. So here's a cool little video. So let me just quickly explain the video. So we're looking top down on a cell experiment. On the bottom of the plate, which we are looking straight into, are endothelial cells. Now, flowing across, the really bright things are neutrophils. And what they've done is they've treated it with an inflammatory signaling molecule called histamine. So we can see these neutrophils are flying past, and deep in the background, these ghosts are endothelial cells. And the neutrophils are flying past, and over time, we see the expression of adhesion molecules. The neutrophils now, but in response to the inflammatory signaling molecule, and the endothelial cells in response to the inflammatory signaling molecules, are now expressing adhesion molecules, which causes the neutrophils to stick and slow down. We can see them doing what's called rolling, which is we see neutrophils slowly roll along the blood vessel and eventually stop. We can see a neutrophil stop. And once it's stopped, it's ready to migrate into the tissue. But that is an amazing cell experiment to visualize adhesion going on live. Let me just jump. Now, what happens next is chemokines. Now, Chemokines are a subgroup of cytokines. So chemokines are cytokines. And what chemokines do is that they're positively chemotaxic. So what's going to happen now is a cell will release a chemokine, which means around the cell will be a high concentration of a chemokine, and further away from the cell will be a lower concentration of chemokine. And what will happen is immune cells will migrate in the tissue up that concentration gradient, right? So towards that chemokine. So if we put these two processes together, neutrophil adhesion to the endothelial cell wall, and now it can detect chemokines and want to go up that concentration curve. So it's now going to squeeze between the blood vessel and migrate towards the chemokine secreting cell. We end up with immune cell migration. Now I've got a good video of this. Let me quickly explain. In red, we have damaged cells. So these damaged cells have released DAMPs, and the immune cells around these damaged cells are now releasing cytokines and chemokines. So the red cells are damaged cells, and we're looking down onto, this is actually super cool. So during a tutorial, I mentioned that we did research on zebrafish and people laughed, but we use zebrafish because when they're young, they're transparent. So we can actually look into a live zebrafish, genetically engineer it so the cells fluoresce certain colors, and then watch what's happening. So in this zebrafish, the endothelial cells are blue. So the blood vessels have been made to express a fluorescent blue protein. The neutrophils are expressing a fluorescent green protein, and damaged cells are expressing a red fluorescent dye. This is actually a fluorescent dye rather than expression, sorry. So these damaged cells are red because of a dye, and the blue cells are blood vessels and the green cells are neutrophils. So let's watch what happens in response to the damage-associated molecular patterns that release from the damaged cells, which have activated immune cells within the area. This is so cool. This really gets my goat. Look at the neutrophils. The neutrophils are adhering to the blood vessel. Let's just watch this on loop. Can I loop it? Loop. Let me just loop it. Yeah, cool. Okay, so the neutrophils are coming along the blood vessel. They're adhering to the adhesion molecules, and they're migrating up the chemotaxic gradient to get near an immune cell that's expressing the chemokines. Look at that go. Honestly, I could watch that all day. That is some beautiful research right there. So cool. So this is an immune response happening live in front of your eyes. It is awesome. These are minutes down the bottom. So it has taken about an hour for this to happen. So I'm going to do a full video about the neutrophilic response to SARS-CoV-2, but let me just give you a quick breakdown of maybe what the neutrophils are going to be doing once they get there, because there's some really interesting research around neutrophils. So another cytokine called IL-8 will bind to the IL-8 receptor on the neutrophil, and this will cause degranulation. Now, in those granules are antibacterial enzymes and antiviral enzymes and antipathogen enzymes that will produce compounds that will kill everything in the area. So let me give you a couple of examples. One is neutrophil elastase. This is a very non-specific protease. So it's an enzyme, it's a protein enzyme that chops other proteins, and it does it non-specifically. So it just chops up all the proteins. And this is what we were talking about when the innate immune system is both non-specific and causes tissue damage. Imagine in a late-stage COVID patient, their lungs are full of neutrophil elastase that are digesting up all the proteins, even their own proteins in their lungs. It's not ideal to have this chronic inflammation, which is why some anti-inflammatories, which are kind of immune suppressing compounds, are beneficial in late stage SARS-CoV-2 or COVID-19. Another example is the neutrophil will produce reactive oxygen species, hydrogen peroxide, and this hydrogen peroxide can be reacted with chloride to produce hypochlorous acid, which is bleach. This is chemically identical to bleach. Your neutrophils are producing bleach. Now, crucially, this bleach isn't in the granules, because if it was, it would destroy the granule, right? Because bleach destroys everything, right? And no, Donald Trump, you can't just put bleach in your bloodstream. it will kill your cells as well as the virus. So anyway, sorry, off topic. Yeah, so we get hypochlorous acid. The crucial thing here is it's not in the granules. What's in the granules is an enzyme, myeloperoxidase, or MPO, that produces bleach. So in the granules is another enzyme, and this enzyme produces bleach, and the bleach can kill viruses. So this is just an example of what's going on with the neutrophilic response in SARS-CoV-2. But let me break you down, because I mentioned another cytokine earlier, the interferon. Now, a lot of viral researchers talk about interferon, and it is very, very cool. So there are multiple kinds of interferon, and they do very overlapping things. So I'm just going to talk about interferons as a group that do roughly the same thing. So interferons are a cytokine, and they're released in response of detection of a PAMP. So you detect, your RIG1 is activated in response to viral RNA, and that causes the release of interferons. These interferons then go and bind to interferon receptors, which then cause a phosphorylation cascade, which is how so many receptors work. So basically, this molecule will phosphorylate the next molecule, which will phosphorylate the next molecule. So we end up with a phosphorylation cascade again. And what we end up with is the gene expression changes, thanks to IRF, which is interferon response factors. We end up with gene expression changes that stimulate interferon-stimulated genes. Sorry, I'm blocking the slide. Here we go. Interferon-stimulated genes, or ISG. So we end up with these gene expression changes in response to the receptor activation. Okay, so which cells are expressing these receptors? Which cells are going to respond to the interferon response? And this is a little bit unique, right? Whereas cytokines typically act on immune cells or vascular cells or cells important, inflammatory interleukins, for example, typically act on cells important to the immune response. Interferon cytokines act on all cells. So this is Human Protein Atlas, so we can just type in any gene in here, and this is one of the interferon receptors, and this is the expression level of the interferon receptor. And along the bottom here, we've got different things here. So we've got enterocytes, we've got ciliated cells, club cells, we've got hepatocytes, so those are liver cells, and all the cells are expressing interferons. So it is really a massively important viral alert cytokine that tells all cells in the body, you need to start defending yourself from viruses by expressing these interferon stimulated genes, these ISGs. Let me just compare that, because it's a bit hard to understand what that graph means, to say hemoglobin. If we look at the gene hemoglobin, where is it expressed? Nowhere except for the blood cells, right? Erythrocytes. So that's a good comparison. Interferon receptors are expressed everywhere. Other genes are typically only expressed in one or two or a few cells. So what happens once these ISGs, the interferon stimulated genes, are turned on? Well, when a gene turns on, obviously that causes mRNA, which causes proteins, and these proteins that are now expressed are now produced thanks to the interferon signaling. These proteins attack viral life cycles. So this is a rough viral life cycle. This isn't the coronavirus, but it's a different virus life cycle, but it's all very similar. And each little red star here, you can see these little red stars here, are an interferon stimulated gene protein, so the protein that comes once you turn that gene on, and each of these little stars attacks the viral life cycle in these locations. And there is a load of them, and they attack the viral life cycle in so many points, and that's because otherwise, if we attack it at one point, the virus will evolve around that system. So we need to attack it at every stage in its life cycle as part of our defense. So the interferon response genes, obviously I can't look at all of them, but let's just look at two of them so we can break down sort of what do they do? What do they do? How do we stop a virus replicating inside our cells? So I'm going to look at two, CH25H, which is an enzyme that hydroxylates cholesterol, and ZAP. ZAP sounds cool. Honestly, that's why I chose it. And then I looked at what it did, and what it does is awesome. So it was a good example. But let's go with ZAP. It's nice and easy to remember. So ZAP is a protein that is stimulated by interferon receptor activation. And what ZAP does is it's a zinc finger antiviral protein. It recognizes the CG2 nucleotides, and it detects those regions, the CG-rich regions, and all it does is it binds to it. And once it binds to it, now the RNA is kind of useless, right? It can't, ribosomes can't bind to it. It can't be used to replicate more of the viral genome. So now this protein's bound to it, it becomes useless. And this useless RNA with a protein bound to it then gets degraded, right? So this ZAP protein binds to viral RNA, stops its use, and breaks it down, signals it's breaking down. So if we look at, this is the genome of SARS-CoV2, and we can see all of these CG motifs in yellow. I will say that there is some evidence that the SARS-CoV2 virus specifically has undergone an evolutionary process to reduce it. We would actually expect more CG motifs in this genome than what we're seeing in a viral genome. So there's been some selection pressure to reduce its CG content, and that selection pressure includes ZAP. But in saying that, they've still done experiments to show that ZAP is an important defender in SARS-CoV2. So what we have here is each of these blue circles are human cells in a dish. And when these human cells get infected with a virus, they will turn white. They will release, they will come up as positive on this assay. So we can now see this one cell here has the SARS-CoV-2 virus. And these numbers here are the dilution. So 10 to the negative 9 is 0.000000001 moles of the virus, right? It might not be moles, I can't remember the unit, sorry. But anyway, these are, it's just dilution factor of the virus. So these are very diluted virus inoculations that are then going on the cells. And as we go up the concentration of viruses that we're treating the cells with, so we're treating the cells with higher and higher concentrations of virus to look at the infection rate, we can then map, is the virus more infective in one group of cells compared to another? Now the two rows here, this is the control, and this is a group of cells that have had the ZAP protein genetically deleted from them. So they are knocked out. That gene is not present in these cells. And what we can see is that the viral infection begins to occur at lower concentrations of virus. In other words, these cells are less resilient against being exposed to concentrations of the virus. And we can see here at this concentration, the control group is almost not infected with the virus at all, and yet in the ZAP knockout cells, there is loads of virus. So from this experiment, we know the ZAP is important in defending ourselves against the SARS-CoV-2 virus. At this concentration of the virus, the cells just die. So that's why we see this dip in infection rates. They can't fluoresce yellow. They can't release this. They can't test positive in the assay if they're dead. Right, that's cool. That's how ZAP, which is induced from interferon, protects us from SARS-CoV-2. Now on to the next one, CH25H. Yes, so it catalyzes this reaction here. It takes cholesterol and it turns it into hydroxycholesterol at the 25th carbon. So that's what that number means right there. So it just adds this little hydroxy group to the cholesterol. How could this possibly help protect us from a virus? Well, the important thing to note is cholesterol is present in your plasma membrane. So in the membrane, cholesterol is there. It actually increases the rigidity of that membrane. Ooh, sorry, I've clicked off it. Here we go. Right, so, and there's something that happens. So the spike protein shoots into the host cell's membrane, and it begins to fold, which pulls the membranes closer and closer together, right? So the viral membrane and the host membrane gets pulled closer and closer together. But something needs to happen once it gets to a certain level of closeness. The membranes need to fuse, right? How does that happen? Well, if we go back to first year biology, we'll remember that inside a phospholipid membrane, there is a lipophilic domain. So all these little tails here are scared of water and love fat, so they stick together. And then at the heads of it, we have a phosphate group, which is hydrophilic, and it's attracted to the water. So that's why the membrane self-orientates into the structure. But what this means is that these hydrophilic heads and these hydrophilic heads actually attract each other. Like things attract in these cases, so lipophilic things, oils like oils, and waters like waters, and hydrophilic things like hydrophilic things. So there's actually an attraction between these two hydrophilic domains, and there is an attraction between these lipophilic domains. And this attraction, once we get close enough, takes over and allows the fusion of the membrane. So now we can see these two lipophilic regions are now loving being with each other, and these two hydrophilic regions are loving being with each other. And so we've got this viral membrane fusion with the host. But what happens when we get this hydroxycholesterol in there? Well, the hydroxycholesterol, it seems to reduce the lipophilic nature of this, and thus reduce the difference between the hydrophilic and lipophilic domain. So the difference here between basically how repelled this lipophilic domain is from this hydrophilic domain is, and how attracted this lipophilic domain is to this lipophilic, decreases. Now, I will say this is leading edge research, and so this is our best guess at how this works, but research is ongoing. But there was a fantastic paper that used just lipid particles, so there's nothing else going on, it's purely synthetic, using just lipid particles, phospholipid particles, and they showed that if you put this 25 hydroxycholesterol in, you reduce the lipophilic hydrophilic difference in that particle, which allows fusion a lot easier. So it was fantastic research, so I fully buy into it. So what that means is that the attraction between these two membranes has gone down, because this is less hydrophilic, this is less lipophilic, so this is less similar to that, and this is less similar to that. So now the attraction between these two membranes has gone down, which means that we repel this fusion. We can inhibit the fusion of the viral membrane with the host membrane by including this 25 hydroxycholesterol into the phospholipid membrane of the host. So this is how this enzyme works. It increases the concentration of the hydroxycholesterol, which decreases the attraction between the host membrane and the viral membrane. And here's some research, hot off the press here, guys. So basically, these cells will fluoresce when a viral particle enters them. Basically, the viral particle is fluorescent, so if the virus can get in there, it will cause the cell to fluoresce. Here's the control group, and so this just has vehicle in it, and regular cholesterol. And you can see as you increase the concentrations of regular cholesterol, you do not change the infection abilities of SARS-CoV-2. But here, they've increased the concentration of the hydroxycholesterol. And you can see as we go from 0.1 micromolar to 1 micromolar to 10 micromolar, so as we increase the concentration of 25 hydroxycholesterol, we decrease the ability for the virus to infect the host. These host cells. So these cells will fluoresce when a virus gets inside, and you can see that these are less fluorescent than that, and that's because of the 25 hydroxycholesterol. So we can see with research that came out, a few months ago, that this 25 hydroxycholesterol is super important in stopping the viral particle fusing with the host membrane. And so that's just two examples of how the interferon response can inhibit the viral life cycle, and why interferons are so important in our viral responses. So now just quickly onto resolution. We need this inflammatory response to stop. We can't let it go ongoing, and that's because it does cause those damaging side effects, right? And this is roughly the cycle of how it works. Inflammatory signaling initiates anti-inflammatory signaling. So cytokines and prostaglandins, inflammatory signaling, the release of those signaling molecules will release a bunch of anti-inflammatory signaling molecules. And some examples are glucocorticoids. These are stress steroids. You will have heard of, I imagine, cortisol, which is a stress hormone. And these are released in response to inflammatory signaling molecules. And these are hugely anti-inflammatory. If you have a swollen knee and they want to inject a steroid into your knee, they inject a glucocorticoid to totally inhibit that inflammatory response. We also have anti-inflammatory cytokines. So remember, some cytokines like IL-1, IL-6, TNF are inflammatory. Some cytokines are anti-inflammatory, like IL-10. And remember, IL is short for interleukin, which literally means compound inside of leukocytes, interleukin. And then there are also resolvins, and these are lipids. These are a lipid that are produced from digestions of those lipids. So it's very similar to a prostaglandin. This is the yin to the prostaglandin yang, resolvins, and they are produced by enzymatic digestion of lipids, and in this case it's omega-3. And so this is probably why you've heard taking omega-3 supplements is anti-inflammatory. That research is actually pretty limited. You know, there is limited convincing evidence of the health benefits of omega-3. It's minor, all right? I wouldn't overstate it. I think it's often overstated, but it is true that omega-3 is digested into these anti-inflammatory compounds. It's just probably not the rate limiting thing. You're probably not running out of omega-3 to produce resolvins. Right, so that is the innate immune response. The last video in this video is the innate immune response to SARS-CoV2. Up next in the next video, we have a little thing about, because I like to cover methods, about how do we use antibodies in research. I'm going to introduce what an antibody is and how do we use it in research.